Thank you a lot, Feng, and for inviting me. It's a great pleasure to be here. And thank you for the very generous introduction. So I'll share with you some of work that we have done in recent years on how to leverage discovery by integrating the generative AI statistics and also the genomics health. So we all know ChatGPT is a tipping point for AI. And so right now there are many type of unstructured data we were not familiar with before, and such as text data, imaging data, and voice data. And also data become much more complex and also become more heterogeneous. And so many of the traditional statistical learning theories is challenged by the emerging learning method, and such as a transformer, which is behind the large language models, and also diffusion model, which I'm very passionate about. And also compared to industries, academia face much more challenges in terms of computational resources, such as the number of GPUs. And so how can we find the sweet spot between statistics and AI and the genomics health? So as we know, like compared to other disciplines, biological and health science are more complex and also affect life. So therefore, generates a high bar and for discovery and also for translational science. And so how can we meet this high bar and for the true discovery? And so then the question is, how can we empower the true discoveries by leveraging AI transformative potentials and also rigorous statistical principles? So this talk has two parts. The first part is how can we empower statistical genomics and by leveraging synthetic data generated from the AI models. So we all know like lots of development in the last few years on generative AI. So the major focus of those AI models is on prediction. And those are wonderful, predictions wonderful, but predictions not everything. And so the influence on population parameters, such as the effect of genetic variance on function, and the effect of genetic variants on common diseases are equally important. The reason is it's important to understand the science, and that can help identify the drug target. And also that can help take the actions. For example, if a clinician tell patient, so this is your polygenic risk score, and the patient will say, I cannot change my genome, so what can I do? And so therefore, it's important to think to develop action. So that's why the influence on population parameter and understanding the science is equally important. Then how can we empower the statistical genomic analysis and the true scientific discovery by leveraging the generative AI, such as the transformer and the diffusion models? And so this where the synthetic data generated by AI model could help. So this is a very interesting talk, and which was presented by Lida Sakava as the Europe 2024. So we all know he is a brilliant scientist. He was the former chief scientist of OpenAI. And so he made two interesting points. The first one is about data wall. So because the company generally, they have a lot of resources. They do those kind of trainings of large language models. And so he was saying like the pre-training will end. And so then the data is not growing because there's only one internet. And then what are the directions he's thinking? So the first thing he mentioned is the synthetic data. I was happy seeing he mentioned the synthetic data. I'll illustrate the power of synthetic data. And then the second one is the AI agent. The third one is on inference and the computational inference time computation. So basically the scalability and the cost efficiency. So how can we generate synthetic data? The traditional way to generate synthetic data is using parametric models, such as a linear model, logistic model, and so on. And also, there are non-parametric way to generate synthetic data. For example, random forest, XGBoost, that many of you are familiar with. In the last couple of years, multiple new ways have been developed to generate the synthetic data. One is the variational autoencoder. And so basically, you start from the original data, try to decode it, and generate the latent feature space, and then regenerate the data using the encoder. And so the key assumption for variational autoencoder is the data need to have low dimensional representation. So one can do that through representation learning. However, not all the data have low dimensional representation. And a second way is doing the transformer. And this basically is behind the large language model. So for example, if one have a sentence, how are you? And use the transformer, it can be translated to any languages. And so the transformer has been useful to generate the new and contextual samples. So what are the key assumptions of transformer? A major key assumption is the data need to have some kind of meaningful order. For example, the data need to be ordered in a sentence, or in the DNA sequence. One can have using the DNA sequence to predict many things. So many different transformer-based foundation model have been developed in the recent years. And for example, like Informer. So basically, those are the sequence language models using the DNA sequence and to predict epigenetics functions such as cage site and also expression level and so on. And also, Meta develops Xformer, so basically is a platform and that uses the transformer as a backbone, and one can use to do any kind of former one like. And the third type of model which I'm passionate about is the diffusion model. And so the diffusion model has two processes. One is the forward process, and which doesn't involve training. So basically, if you start from an image and just add in a noise, a little by little, and then until it becomes completely white noise. And so the most exciting part is the reverse process that involves generative process. So basically, one try to gradually denoise the data and from the white noise until one recovers the true image. So what are the benefits? And compared to the variational autoencoder and also the transformer, the data do not need to be ordered or have a low dimensional representation and can generate data with complex distribution and also high dimensional data. So what are the drawbacks? And so one is the computational challenge. And so this is really active research areas. And so for example, like UNet is one attractive approach to speed it up. So how does this work? As I mentioned, the goal of diffusion model is to generate a distribution of high dimensional data like an image, and from that mimic the original data, observed data, or one can use the conditional diffusion model to transfer the high dimensional data. For example, I take a picture of this room. The background is the wall. I can change the background to any background you like. So diffusion model has been used to generate many kind of images, and we are familiar with in the softwares. And so to show you an example, suppose the true data is the bimodal normal distribution. Then the forward process is you add in the noise until become a completely white noise, like normal distribution, and reverse process you regenerate it, and then it become bimodal distribution again. Then how does this work? The reverse process. The reverse process basically is the denoise process. So you start from the white noise, gradually remove the noise, estimate the noise from the data, gradually, little by little, until we recover the true image. So how can one denoise it? By estimating the noise. That is where the learning comes from. So basically, one can train one can predict the estimated noise by predicting the white noise and using the data at any step xt to predict either the white noise or predict the score function, sun score function, and through the score matching. So then the question is, giving those powerful synthetic data tools, and then how can we empower statistical genomics? As you probably know, like right now, there are lots of interest in virtual cell, and virtual cell basically is a generative model. And so how can we empower those kind of synthetic data from generative AI to empower statistical genomics? I'm going to illustrate the point and using one paper which we published last June and in Nature Genetics from using the GWAS study. And so that is my former student, and this is his third dissertation paper. Jessica is our former alum. So the question we want to answer is in UK Biobank, for example, those biobank data. So generally some high quality phenotype are very expensive to measure, such as body composition. So they are only measured by a small fraction of people. And so given a large fraction of people don't have body composition, and then how can we use the synthetic data generated by AI and to empower the GWAS analysis? To illustrate, and so the body composition from UK Biobank is measured by 30,000 people. And so we all know UK Biobank has 500,000 people, so that means 95% of people body composition is missing. And so that is represented by the orange Y. And so, but we have a lot of predictors, such as demographic information, biomarker, ST10, ST9 code, which are available for everybody and in 500,000 people, but restricting to the European independent sample, there are about 350,000 people. So these are marked by blue S. And we want to do the GWAS analysis. So X indicates the genotype, ancestry, PC, and so on. So the goal is to run the GWAS of the Y body composition on the SNP X. But 95% of body composition is missing. So what can we do? So let's predict the body composition by using the surrogate demographic information, biomarker, and ST10, ST9 code. How can we do the prediction and generate the synthetic Y hat? So one can do multiple ways. One is that suppose you have a pre-trained model and using external data, this can come from either random forest, transformer, diffusion model, where you have the weight. And then I'm going to take the weight and fine-tune it and apply to the UK Biobank data, and then to generate the Y hat. So that is one way, and through the fine-tuning of external weight. The second is you split the data and use a part of it UK Biobank data to do the training, and then part of the data to do the analysis. Then suppose you use the, we use the 5,000 people and to train the model and to predict body composition. And then I'm going to have because I observe biomarker, demographic information, ST10, ST9 code for everybody in UK Biobank. So I will be able to predict body composition for everybody, for 500,000 people. That is marked by Y hat, the red. And then we can use those, the Y and the Y hat together to do the GWAS analysis. Then the natural question to ask ourselves is that how can we ensure the GWAS analysis is still valid if my black box AI generated model is wrong? Suppose my Y hat is a garbage. How can I, if I just run the GWAS analysis of Y hat on X, if Y hat is a garbage, then you know the GWAS analysis result will be garbage, right? And also then how can we ensure that GWAS analysis is still valid if my AI model, generated model is wrong? So what are the analysis strategy? The first strategy is just do the standard analysis. So basically, I only use the label data to run the GWAS. I have 30,000 people with body composition. I run the GWAS of 30,000 people, regress Y on X. Second, I can do either single or multiple imputation. So I basically impute the missing Y and then run the GWAS on Y hat. So you can see I have 30,000 people with observed body composition. The leftover without observed body composition, I just run the imputation model, generate the y hat, and fill the missing value. This is the traditional approach. And then if you just look at this, you can see my imputation model had to be correct in order for this approach to work. So, but if the third approach is this synthetic approach I'm going to talk about, that means that I'm going to analyze the jointly of observed body composition for 30,000 people and the predicted body composition of 500,000 people, and then do the joint GWAS. And so this will try to calibrate, even though my AI generative model is wrong, my y hat is garbage, it doesn't hurt me. So this is ensure positive learning. So how can we do that? And so basically, suppose we have used 5,000 for training, I have 25,000 left over for analysis using the observed body composition. I have a y hat for everybody. And then we can do the joint analysis of the y and the y hat on the GWAS analysis, basically treat y hat as a phenotype as well, but also y is a phenotype, but y has a missing value. And then the covariance matrix, the sigma TS, basically measure how good the generative AI model is. If the generative AI model is good, then my sigma TS will be close to 1, basically the correlation is 1. And so this turns out we don't need to fit this model. We can run simple univariate analysis sequentially. We don't need the special software. So how can we do that? So first, because I predict body composition for everybody, I'm going to run the GWAS analysis using the predicted body composition as my y hat, as a phenotype regress on X. And then after I do that, and I got the residual. And then next step, I need to calibrate. And so basically what I do is we use the observed body composition for 25,000 people, regress it on the residual from the first step, and then also regress on the genotype. So therefore, you can see even my y hat is the garbage. It doesn't hurt me because I regress it on the E. So that is try to calibrate. So the coefficient from this second stage and basically give me the same coefficient as the joint GWAS analysis. So basically, this is a schema. So I use the first 5,000 people to train the model, and then use the leftover data for analysis. For the training, you can do any kind of the machine learning model, either using the internal trained data model or use external weight. And so neural network, whatever you like to use, then you got the weight. You apply the weight to the rest people in the UK biobank sample. You apply the weight to everybody in the UK biobank sample, then you do the joint analysis, which can be done and using the sequential approach. You basically treat observed body composition and predicted body composition, both of them as your phenotype. And then you have the GWAS Manhattan plot. So what if my generative model is wrong? And so you can see if my generative model is correct on the left-hand side of the figure, and the standard analysis basically using observed phenotype. And then the oracle is a situation, suppose you observe everybody, the body composition for everybody, and then the blue and the red is a single and multiple imputation. Purple is the synthetic approach. You can see if the generative model is correctly specified, everybody's okay. Everybody's unbiased. However, if my generative model is wrong and the multiple imputation will give you biased result, and also observed data will give you biased result. But if you use the synthetic approach, you can see that still give unbiased result. So does this work? How does this help? So we look at the standard analysis. For example, look at the lag mass. This is the number of GWAS discovery. And so if we use the 30,000 people, because you can see the sample size is too small, we don't see many discovery. The number of discovery is the red. And if we use the synthetic approach, you can see you have much more number of discoveries. And then the natural question is, how do we know those discoveries are the true discovery? We have to benchmark it. So we benchmark it by comparing the number of the discoveries with the SNP in the GWAS catalog. And so you can see the red is the number of overlap of using the standard GWAS analysis with using the UK Biobank data with the GWAS catalog. You can see the overlap is not much. But if one uses the synthetic approach and the GWAS approach, you can see the overlap is quite good. And the orange basically is the difference between the blue and the red. That indicates the number of the validated discovery. So this is quite encouraging. So the benchmarking is critically important, showing this approach work. And then one may wonder how much efficiency gain can we get, statistical efficiency gain. Basically, this affects the power. So this the statistical efficiency gain basically compares the variance of the GWAS GWAS effect estimate using the complete data, 30,000 people versus the analysis beta hat variance using the GWAS result using the synthetic approach. And so you can see that is a function of the proportions of missingness and also how good the generative model is. The row indicate how good the generative model is. So give you example. Suppose 99% of the body composition is missing in the UK Biobank. And suppose my generative model is perfect. So the correlation between the Y and the Y hat is 1. Then basically the relative efficiencies, this is a statistical relative efficiency is 100. So that means you reduce the variance by 100 times. That basically means you effectively increase the sample size by 100 times. So therefore, if you have more missingness, if your predictive model and the generative model is really, really good, and then you can effectively increase the sample size a lot. So suppose that your Y hat generative model is completely wrong, it's a garbage. So the row is 0. You can see this relative efficiency in the last column is still 1, and also there's no bias. So it doesn't hurt you, even your generative model is a garbage. However, if your generative model is predictive, and suppose your row is 0.75, that basically R square is 0.5, you can see the relative efficiency is 2. That means you double the sample size. So, sorry, I think the title is missing. And so when I give probably some of you are familiar with this predicted power inference. This was published in Science probably in the fall 2023. And then so this is by Michael Jordan's group. And then how the synthetic approach is related to the PPI. And also after our paper was published, there was another paper called PubGWAS, and it was published. And so I'm going to look at how those methods are related. So this work was led by my postdoc, Roy Wang. So let's start from the PPI and this in the Science paper. And so basically, this is a very nice paper. So the idea is, suppose you use the observe the data, like 30,000 people with body composition, you do the GWAS on X, and you have the beta hat. Okay, that basically regress orange on the green for 30,000 people. And now suppose you have a labeled data that 30,000 people, you have Y hat, and you regress the predicted body composition on the SNP. Then you get alpha labeled. Okay, and then alpha hat unlabeled, basically you have the predicted body composition for the rest of the GWAS sample, you regress it on the SNP, and then this is the blue, alpha unlabeled. So this PPI method has very simple formula. So basically you use alpha hat labeled minus alpha hat unlabeled. Those are the GWAS result using the predicted body composition. And then take that difference, and then you use the beta hat, subtract this difference. You can see the estimation is very, very simple. So by looking at this formula, you can see if my alpha Y hat is bad, this doesn't calibrate. And also, if my labeled data and unlabeled data are different, then you can see this will be biased. So then the PubGWAS try to calibrate it. So basically multiply the coefficient of that difference by a constant. That constant is basically the correlation of Y and Y hat to measure how good the generative AI prediction is. And so this will improve over the PPI. So among all the three approaches, And so what are they in common? They are all robust under the, when the predictive model is misspecified under missing at random, missing completely at random. What that means is basically the labeled data and unlabeled data have the same distribution, and then they are unbiased. However, there are multiple differences. The first difference is both the PPI and PUB-GWAS, they are not statistically efficient. Basically, they have larger standard error and also lower power. However, the synthetic approach is statistically efficient, has the smallest standard error, and also the highest power. Second, there's an issue with the negative learning. So the standard error of a PPI can be bigger than the standard approach, and if the predictive model is misspecified, so that is not good. So if you have predictive model, suppose the correlation is R-squared is 0.5, and your standard error using this approach can be bigger than just using 30,000 people. So there's a negative learning here. However, using the synthetic approach, I can guarantee the standard error using this synthetic approach, GWAS approach, is always smaller than the standard error just using the 30,000 sample. There's no negative learning. And even the predictive model is misspecified, is garbage. And also both PPI and the PUB-GWAS require the data to be missing completely at random. So that means the labeled data and unlabeled data need to have the same distribution. However, the synthetic approach only require missing at random. So that means I don't need the distribution of the labeled data and unlabeled data to have the same distribution. So to show you, so let's start from missing complete at random. That is the labeled data and unlabeled data have the same distribution. So the row indicate how good the predictive model is. The row is 0.8. So you can see the R-squared is almost 0.5. It's pretty good, the predictive model. So in the left, is the orange is using the standard approach, just using 30,000 people. And the green is using the PPI, and the blue using the PUB-GWAS, and the purple is using the synthetic approach. You can see all the estimator, when the labeled data and unlabeled data have the same distribution, you can see they are unbiased. However, you can see that the standard approach and also the PPI and also PUB-GWAS, they have a larger standard error than the synthetic approach. Furthermore, and if you look at the PPI standard error, and it can be even bigger than the standard approach. So that means there's a negative learning if the predictive model is not 100%. So it may hurt you. And so if one look at the missing at random, that means when the labeled data and unlabeled data have a different distribution, and in this situation, you can see all the other estimators are biased, but the synthetic approach and still ensure the unbiasedness. So that is good. So to summarize this part, and so the AI generative synthetic data are powerful and with many potential. Here we only illustrate using this missing data example. So it leverages the AI trained synthetic data to empower statistical genomic analysis with missing data. So with many of advanced technologies, and those technologies are expensive, then if we can generate good synthetic data, you can see if we do the right synthetic analysis, and then that can empower analysis. And so this offers a more robust and powerful analysis than both the PPI and the PUB-GWAS. So basically, we need to ensure the positive learning. So when we do the AI generative model to generate synthetic data, if we don't do the analysis right, it can hurt you. It can have negative learning, but we try to avoid negative learning. So therefore, by bridging statistics and generative AI, it can empower us for true discovery. So the second part is I'm going to talk about whole genome sequencing analysis, give you overview of the whole genome sequencing analysis, to build this ecosystem, end-to-end scalable whole genome sequencing analysis ecosystem. So in order to do that, we will need both the statistics and also machine learning, informatics, and computational biology, and also some engineering. So to do the whole genome sequencing analysis, we need to first functionally annotate the genome. And then we want the analysis pipeline to be fully automated. And also because right now whole genome data cannot be downloaded, they are all on the cloud. So therefore we need to deploy it in the cloud and also make it affordable. People can afford it. So in the last couple of years, and the sequencing costs have dropped dramatically. There are several large whole genome sequencing programs. One is the TopMed. So we have sequenced about 200,000 people. The other one is the genome sequencing program. We have sequenced about 140,000 whole genome. And so there are multiple large biobanks. The most well-known one is the UK Biobank, which has the both the GWAS data and the whole genome sequencing data of 500,000 people, and also with the electronic medical record and also epidemiological data. Another one is a Million Veteran Program and has enrolled 1 million people. They are doing the sequencing now. And the third one is ALPHAS. Basically, this is the US version of UK Biobank and has enrolled 1 million people. So far has sequenced about 400,000 people. And so in the last couple of years, there are lots of effort on pushing for data fairness. So you can see all those large whole genome sequencing studies have multi-ancestry sample. And so TopMed and GSP have a 40% European, 30% African American, and 16% Hispanic, about 10% Asian. ALPHAS has a 50% European, 18% African American, and 17% AMR, about 3% Asian. And so when we analyze those data, we need to account for population structure and also for relatedness as well. So to show you how big the data are, so this is a TopMed Freeze 10, has about 185 whole genome. Before QC, there are about 1 billion variants, and so many of the variants are rare variants. So for example, after the QC, there are about 845 million variants, about 45% are singletons, and 16% doubleton, only less than 1% are common variants. So basically, after we finish the sequence the genome, that's pretty it. That's just a whole genetics code. But then how to read the code is a challenge. So the goal of whole genome sequencing analysis, if we want to scan the genome to identify SNP site associated with the diseases and trait, because the variants are too rare, and then we cannot do single SNP analysis like in the GWAS study, but we have to group them in a set. So that is where the art is and also where the challenge is. And to do this, we first need to functionally annotate the genome. So for the GSP, we have this genome sequencing program annotation working group. And so we integrate many functional data across many different sources and build the FAVOR functional annotation database. So we build it for all possible variants. Across the genome, they are about 3 billion positions. Each position, there's 1 reference allele, 3 alternate alleles, so totally 9 billion variants. Right now, we observe probably over 1 billion. So there are many variants which have not been observed, but if we continue increase the sample size, we are going to observe more and more variants. And so we make this database public available. This is a PostgreSQL relational database with very efficient indexing. So if one wants to search one particular variant, it takes like a second. And then we include both allele frequency, conservation, protein function, epigenetic function, and from ENCODE, and also we also include like alpha missense, many different annotation. Any of your good annotation, we will be happy to include as well. Everybody is familiar with computational biology, so we want to make it user-friendly. And to do that, we built this FAVOR GPT interface. And so that provides a large language model interface and use the FAVOR as a backend database. And so we built the FAVOR API, and using the ChatGPT API, that creates a nice interface. And so this is led by several members in my lab. So you can do any kind of query like what you do in ChatGPT. For example, you could ask, what is the function information of this particular variant, which we all know this is a well-known variant in ApoE. Then that will use the FAVOR backend database and to do this, the query, and then summarize the finding. And so you can do all kinds of calculation. For example, one can ask, so how many pathogenic variants does BRCA1 have? Then because FAVOR includes the ClinVar database as part of the database, then it tells you 92. However, if you ask the same question using ChatGPT, it will tell you 1,000 because there's a hallucination problem we are all aware of. And so this for the general, it's good for general query, but if one need to have some kind of specialized query, one need to have a strong professional database. And so the CleanMark is 92. And so, as I mentioned, our goal is to make the whole genome sequencing analysis as an ecosystem and end-to-end ecosystem, make it scalable and also affordable and also automated everything. The traditional way to do data sharing is basically the data copy. So the UK Biobank data, in the old day, we could copy to each our own clusters, and then that basically bring the data to researcher. And nowadays, because the data are too big, and so UK Biobank doesn't allow the data to be downloaded anymore. Everything is on the cloud. So the new model is to bring the researcher to the data. And there are many different clouds, like Google Cloud and Amazon Cloud. So in order to do this analysis and deploy it in the cloud, we need to do data science engineering. To show you a few additional examples, the data only on the cloud. One is the GSP data and all-fast data, and they are on Anvil. And also, Feng mentioned IGVF. We are going to make the data available, IGVF data. This basically think about IGVF as a new encode. And so we will make IGVF data available and in Anvil as well. So those will be all on the cloud. And so the whole genome data of all of us 400,000 people cannot be downloaded. And for TopMed, the TopMed data, 200,000, the whole genome, they are on Biodata Catalyst. And this is GLBIO data common. And for UKB data, they are on RAP, and the whole genome data cannot be downloaded either. So therefore, we need to make everything, whole genome sequencing analysis done in the cloud and make it affordable. So you may wonder how big the data are. So for UK Biobank, 500,000 people, and the data are stored in the PVCF files. There's 150,000 files. And so you can see that totally the file size is about 1,500 terabytes. 1 terabyte in the Google Cloud is cost about $300 a year. So if you use the 1,500 times 300, you can see that's quite expensive. You just store the data in your own cluster without doing anything, and none of us can afford it. So the first thing we did was we tried to create a compressed data that can be read into R for analysis. And we created this called AGDS format. And so GDS file is used for gaming. And so AGDS basically is annotated GDS. So basically we put annotation and also the genotype file in a single file. So you can see the compression rate is really nice to see. It's about 1,400 times smaller than VCF file. It's only 1 terabyte. And so this is much more manageable. And so how can we make this whole analysis pipeline automated? So we put this Fever backend annotation database in all the cloud platform, the Anvil, Biodata Catalyst, and also the UKB. And then one can, for suppose you want to analyze UK Biobank data, one can do the QC. For example, for the all-fast, the QC is done using Hill, developed by my colleague Banyo at Broad. And then one can run the Fever annotator, and to annotate any whole genome sequencing data, we can use to annotate the UK Biobank data that is about 1 billion variants, and annotate all-fast data is about 1 billion. But those 1 billion and 1 billion are not the same 1 billion because they are different samples. So that's why we need to functionally annotate all 9 billion variants, and so that can be used to annotate any whole genome sequencing data. So that's generated annotated GDS file. And then you have a phenotype data from, for example, from electronic medical record, and then one can run the STAR pipeline that will perform both the GWAS analysis and the whole genome sequencing analysis, accounting for population structure and the relatedness. And so then how to do that? So we developed this STAR pipeline, which was published in multiple papers, and 2 in Nature Genetics, 1 in Nature Method, and 1 in Nature Computational Science, and led by my 2 former students. So basically, the idea is we want to empower real-variance association test by dynamically upweighting the variance with a function. And so because when we talk about functionality and each variance, we cannot say it is function or not. We need to talk about whether it's a function in what, whether it's a protein function, epigenetic function, and so on. So we need to, so that's why we do the functional annotation using multidimensional functionality. So we need to group the variance. So for example, group the real variance into loss functional variance or disruptive missense variance or synonymous variance for the coding variance. For non-coding variance, we can group them into the promoter or the enhancers. And then we can also do the sliding window approach with either the fixed window size or dynamic window size. So coding regions are easier to analyze than non-coding region, as we all know. So because when we talk about functionality of non-coding regions, and they are tissue and cell specifics, that's why the IGVF was launched to try to understand the functionality of the variants, especially for non-coding variants at the context and cell level. So this is a STAR pipeline. First, we prepare the data, the genotype data, create a sparse GLM matrix to account for relatedness and population structure using the ancestry PC. We run the favor annotator to annotate the whole UKBIO bank data or top-mat data, and then we create a different APC, annotation PC, APC epigenetics, APC conservation, MPC protein function. Those basically are principal component of the different annotation scores. And then we group the variants into different masks. For example, group the variants, think about ApoE, group them into the loss functional variants, missense variants, and promoters. And then we run the STAR analysis, this real variance association analysis using different annotation as a weight. And then by the end, we create association p-value of missense variance of ApoE and LDL, and the association and the p-value of the loss functional variance of ApoE and LDL, and so on. And so when we apply this to the top-mat 66,000 whole genome sample for lipid analysis, all the right lipid gene pop up. So for example, loss functional variance, missense variance of LDLR, ApoE, ApoB, and PCSK9 and PCSK1L1 associated with LDL. We already know that the PCSK9 and also NPC1L1, there are already 2 drugs targeted toward this gene and approved by FDA. And also for promoter, enhancer, LDL, and R and the PCSK9 all pop up as well. They are associated with LDL. So these are all good findings. Then you may wonder how much it costs. It turns out we made it quite affordable. And to run the favor annotator for a whole UKBIO bank sample, 500,000 people, and there's over 1 billion variants, it only costs $6. You only need to do it once. And so then you can use that for any phenotype. And for fit the null model, it costs 25 cents to run any whole genome scan. And so only total will cost less than $10. So basically analyze any phenome, any phenotype in UKBIO bank for 500,000 people, it costs $10 on average. So we make it affordable so people can afford it, run it on RAP. So a natural question is that when we did the STAR paper, we used the global epigenetic score to do functional annotation, basically from the ENCODE. And so, however, when we talk about functionality of non-coding regions, and then they are generally cell tissue specific. So the question is whether the tissue-specific annotation can help us to improve the power. The answer is yes. For example, for lipid, we know the right tissue is liver. So you can see the number of discovery using liver that is marked by red. It has more discovery if we using the heart or the brain as annotation in STAR. So that is good. However, we all know the tissue is a cocktail. So it mixed up with multiple cell type. The most relevant cell type of LDL we know are hepatocyte and adipocyte and so on. So the natural question is, can we move one step further? And using the cell type specific functional annotation to empower non-coding region reference analysis. And so this is where the cell star come from. So basically we integrate the single cell data and functionally annotate variants at the single cell level, and then create a cell type specific annotation and combine it with the star. And so this work is led by my postdoc, Eric Van Buren. So this is how the cell star work. First, we prepare the data like what we do in the star situation, and then we annotate the variants for each cell type. And so you can see we have different cell type, and to annotate the variants function at different cell type. So we use the single cell ATAC-seq data using the catalyst developed by Bing Ren's group. Is a wonderful resource, but it only has a single cell ATAC-seq data right now in that database. So IGVF will generate multiple genome data, including the perturb-seq data and the multi-genome data of single-cell ATAC-seq data, single-cell RNA-seq data, and PRA data, and so on. So when the IGVF data, we will make the data probably available next year, right, Feng? So hopefully next year or two, we'll make the data publicly available to the community. Then we will have more functional data and to do better annotation. And so then we use catalyst data to functionally annotate the variants at different cell type level. So then the question is, after we annotate the variants function at different cell type, mapping elements, regulatory element to gene, this is another challenge. And so there are a lot of uncertainty on mapping the element to gene. Then how can statistics help? So we developed this omnibus real variance p-value test using the ACAT to aggregate different element to gene linkage method. So here we can use the distance-based linkage, we can use the ABC linkage or EpiMap or the screen. As long as one of the linkage show the association of the mask of that mask and LDL, then the ACAT method will pick it up. So basically the ACAT method works as follows. Suppose I have the p-value of this the mask, the elements, and to gene and the linkage. I look at the association of the variants in that elements and the LDL. And so we got that p-value, for example, using the distance method or using the ABC. And we have two p-values. And then how can we combine them? Because you look at the data multiple times from the same data, we need to take that into account. Multiple look at the data, that's where this ACAT method to come. So basically, this is a wonderful way to combine the correlated p-value. The basically is similar to the minimum p test, but account for multiple look at the data and also the p-value are correlated. And so basically, this can be used to combine correlated reference p-value using different linkage methods. So this creates a robust method and for studying the association of elements and the phenotype. And then by the end, we create a Manhattan plot for every for different cell type for each phenotype. To show you the results, so this is the top-match cell star result for LDL using hepatocyte. You can see all the good genes pop up. For example, ApoE, LDLR, and also PCSK9 elements are associated, regulatory elements are associated with LDL. And also, it's good to see that if we use the liver relevant cell type and in the cell star, you can see the results are enriched. So that is also encouraging as well. So to final remark, so we all need to be open-minded and think out the box when we're embracing AIs, and also we want to have a true discovery. And by empower statistical genomics using the synthetic data, and by leveraging the AI generative model, and also account for the possibility that those black box AI model can be wrong. But when the the statistical analysis still valid, and also if AI model help, then I'm going to have a better power for the discovery. And also build end-to-end data science ecosystem for whole genome sequencing analysis, make it affordable. And also we need to continue our effort and to integrate statistic, AI, genomics, and to accelerate true discovery. Thank you.
There we go. Thank you. That was such a fascinating talk. Thank you so much.
Thank you.
A question about your earlier topic on the syntactic data. The example you showed to us was at 30,000 individuals that was observed, and you said that you trained on 5,000 of them. Do you have any idea of whether we have a threshold on how many can we impute compared to how many instead we have? Is there a lower level threshold of observations that below that threshold we shouldn't really even try to make synthetic data, just because we don't have enough observation to begin with? And even if your method is unbiased, you still wouldn't get anything particularly useful out of it.
That's an excellent question. Definitely, this is very much open field. There are many challenges, many opportunities. So right now, I think if you think about just use IGVF as an example, okay? We cannot functionally do the experiment to study the function of every single virus. There's no way, right? Just too expensive. So therefore, like even for IGVF data, we can only study a limited number of cells and a limited number of variants. And so therefore, by doing experiments, there's no way we can recover the function of all the 9 billion variants across the genome. So that's why this community is important. So we all need to work together, and by leveraging limited experimental data, powerful statistical method, and powerful computational biology method, and try to make it scalable and to across the genome. So I think that this basically had to be the way to go.
Okay.
Make sense?
Yeah. But however, in that particular publication, you didn't have any kind of like, have you tried to train into lower and lower and lower thresholds of observed data and then see what happens when you then create the synthetic data?
Yeah.
Do you do any?
Say that again. Oh.
Sorry. You said that you trained of 5,000 individuals.
Yeah.
And that you had 30,000 observed.
Right.
You trained of 5,000, and then you predicted on the entirety of the UK biobank. Did you try to do sensitivity analysis that you kept removing observed patients?
I see. You are saying how to split the 30,000 people, part into the training, part into the analysis?
No, mostly try to simulate scenarios in which your observed data keeps decreasing, and see what happens as data keeps decreasing.
Yeah.
Like in this case, you had 30,000, but say we only had 1,000 or 500.
Yeah. Great, excellent question. Yeah. So as a matter of fact, we just take the 5,000 as a convenience. And but when I was at another conference yesterday, one of a colleague told me that he developed this kind of cross-validation approach to select the optimal split. And so you suppose whether maybe 5,000 is too big or too small. We don't know what is the optimal sample size usually used for training, what is optimal sample size usually saved for discovery. And he said he developed method one can use the cross-validation to select optimal size and for training and optimal leftover for discovery. And I haven't read that paper. I thought that is a good idea. So I think similar to what you are talking about, basically how to balance it, right? Yeah, how to balance it. So if you make the training data too small, then your prediction model R-squared too bad, right? So you cannot make the training data too small. But also if you make the training data too big, you don't have much left over for analysis. So that is a sweet spot you need to find.
Yeah. Thank you.
Yeah.
First off, thank you so much for the great talk. Really like it. I got two questions. So the first one, you talk about empowering the statistical genomes using synthetic data from the AI or ML model. So from my understanding, those generated data are supposed to, the distribution are supposed to be the same from the training data. So the generated data, they are not out of sample data. So, and you show that the performance could be improved from those newly synthesized data, right? So what do you think is the reason for the performance improvement, even though we're using the, I don't know, in-sample data set?
Oh, compared to external data?
Yes.
Yeah, we haven't tried. I think generally it will be good to leverage external data as well. And so basically you can fine tune it. Suppose you got the data from another sample, and then you got the weight and you transport it and fine tune it using the UKB data. I think we have not tried that. And so that would be a good thing to try and to use external data. But I think if one use external data, I think it's more likely that the labeled data from external data will be different and from the in-sample data. So therefore, if one apply the PPI or PubGWAS, one will have a problem. But if one use this and it would be using the synthetic approach, I think one would be okay.
I see. Thank you. And the second question is about the Favor GPT, the tool you developed. So you were mentioning that this Favor GPT can reduce the hallucination, which is great. But I think when people apply those GPT methods, especially in computational biology, it is very important to confidently evaluate what are those generated results, like what are the responses. So how do you measure the the quality of the generated response in the
excellent question. So we all know ChatGPT make up answers, right? This hallucination problem is important. And so I think so it will be there's a lot of effort right now try to create uncertainty of the ChatGPT answer, and for example create a confidence score using the conformal inference. And so that will create sort of like a predicted uncertainty. So I think that that is another example that a statistician and computer scientists need to work together. Yeah.
Okay, sorry, I got one more follow-up question.
Yeah, probably.
Is it okay? I can talk later, maybe.
Okay.
Thank you so much.
Hi, very inspiring talk. Thank you. I was wondering in the SynServe project, with the 30,000 phenotype samples, you have lower statistical power, as you described. So what I'm wondering is, how much is the increase in the number of variants detected with the imputed data simply a function of increased statistical power, and how many are new variants that you wouldn't have gotten had you simply reduced your significance threshold when you do just the 30,000 validated samples?
So the significance threshold should be measured by the how many comparisons you make. For example, in the GWAS study generally is use the Bonferroni threshold. It's nothing to do with the sample size. It depends on how many SNP you do the analysis. So for example, standard GWAS analysis, you use the 0.05 divided for the number of variants, like a million, and that decides the threshold.
Sure. My question though is, if you scaled the 0.05 based upon the sample size that you're actually using, so you increase that for your
I see what you mean.
30,000.
Excellent question. Yeah, I see what you mean. Sorry.
That's good.
You are definitely right. So because you increase the sample size, you can make everything significant, right? Yeah. Exactly. So that's why functional analysis is important. And one cannot just fully rely on statistical significance. And so even you can make things very, very significant, but if it's really not functional causal variance, it will not be that useful. So therefore, the integrating functional information and also statistical analysis is important. You're definitely right. Yeah.
Thanks. And so that means that with the imputed data, you're not only getting better statistics, you're also getting additional novel variants that you would not have seen at all with the different threshold. Thank you very much.
Yeah, definitely. Yeah.
This is a really, really interesting talk. It reminded, so the synthetic data part reminded me a little bit of like the Model X kind of like conditional randomization stuff. But I think if I'm remembering right, I think one issue with some of those tests is that they're not as robust to misspecification. And so I'm wondering if you thought about using this approach in that framework, kind of like something similar to what you were doing, looking at both the kind of like the real and the sort of like error from the imputed phenotype together. If you could do something like that to look at more of like a conditional randomization.
Fully
test
agree.
framework.
Yeah, there are
Cool.
so many directions. Fully
Yeah.
agree what you said.
Cool.
Yeah, because I just illustrate using one example.
Yeah,
but
yeah, yeah,
just
sure,
the
sure.
synthetic data,
Yeah.
I feel is very powerful.
Yeah.
And just have many different dimensions, especially in genomics, because experimental data are limited. So if we can really generate the virtual cell data and then leverage observed data with the virtual cell data, allow the virtual cell generated model to be wrong, and we can have really good discovery, can scale it up. I fully agree with your idea.
Cool. Thank you.
Yeah.